All right, hello, welcome back to another episode of Biology with Miss Chu. So today's lecture doesn't have any associated notes, so it is kind of supplemental in that sense, but it is really important if we want to understand how things like genetic disorders work, because they are based on mutations, which are changes in the DNA. And so that's what today's lecture is going to be about. And I've started off with some examples. And I do want to say, because I always get asked this, these pictures are all real photos, so they're not Photoshop. These are all real mutations that can happen. They are definitely some of the more extreme ones because I think that's more fun. But yes, they are all real mutations. And so we can think of mutations as when things go wrong, but we'll see in a bit that it's not always necessarily a bad thing. And some of them can be quite benign. For example, this cat over here is what we call a chimera. And so the DNA actually in the two halves of its body are totally different. So it's almost like two individuals have been kind of smushed together to make one cat, which is why they are called chimeras. And then this is a bit more dangerous, I guess you could say. So this does happen occasionally, which is cows will be born with two heads. These calves often don't live very long because there's usually other issues with their body structure that are associated with this. But it's quite interesting when it does happen. So here's some more examples. So this one is obviously fictional, but for those of you who play Pokemon or know of it, right? So you could have your regular Pikachu, and then mutations are just ones that are, different from the normal. And then here are some more mutations that can happen in real life. And so again, here's a cat, and you can see it actually has extra toes on its paw. And so instead of 5, it has this extra 6th one, and that's called polydactyly. And so that occurs also sometimes in humans as well, although it seems to be more common in cats. And then here is a mutation that sometimes occurs in flowers where they have just, overgrowth of petals, and then the center portion is also quite large. Here are some more mutations. These ones are maybe a little more gross. Again, the two-headedness occurs in snakes pretty frequently as well. Frogs are an interesting case because they are amphibians, they have this very permeable skin. So it's meant to let in water to keep them moist. But it also means that they quickly will absorb any toxins or other chemicals in their environment. And so if there are chemicals in their environment that can induce mutations, frogs will often show this the most severely in an environment. And so you can see this frog has a bunch of additional legs. This is what is commonly called, I think, Cyclops syndrome, and this is actually a goat. And so, as you can imagine, Cyclops syndrome is when the head or the face does not form correctly. And so it actually creates this sort of one-eyed situation. And this is pretty freaky, I will say. And these, again, they typically do not live for as long because of the associated issues, with the brain development and whatnot. Okay, so those are some examples of, like I said, pretty extreme mutations. But what is a mutation? So very simply, it's a random change in the DNA sequence. And so here's our double helix of DNA. And a mutation would just be anything that happens to it that would change it from what it originally was. So here's an example, right? We could take just one letter in the sequence, right? So we have our different bases here, and we could change it. For example, we take this T, and then it gets changed to a C. All right, and so this could have many different effects depending on where in the sequence it is, what gene it is, etc. And so we'll take a look at that. But first, let's think about where do mutations come from? What causes mutations? So some mutations may be inherited from the parents. And so this would be when the parents' gametes, so they're either their egg cells or their sperm cells, something goes wrong with the DNA in those cells, and then the child is then born with some kind of mutation. And so this might be color blindness, sickle cell anemia, or something even as benign as blue eyes. Those can be passed down from parent to offspring. But there are also mutations caused by environmental factors, and these are ones that are kind of more sensationalized, perhaps, in the media. And so you might have heard of any of these. For example, radiation. So UV radiation from the sun is one of the #1 causes of mutation. And then, of course, X-rays less frequently. Certain chemicals, carcinogens, right? In cigarettes, for example, there's a lot of them. But there might even be ones found in food, right, with certain preservatives or with certain, cleaning products or other things like that. These chemicals, when they come in contact with the body, then they can induce these mutations in the DNA. And then we also have infectious agents. So the damage done by viruses or bacteria to your cells could also cause mutations. So those are some of the causes. All right, and so what happens essentially is so either you have these exogenous causes, right? And so these are more of like the larger scale, abnormal kind of things, or endogenous. These are more things that are found inside the cell. Okay, but whatever it is, the cause is going to either break the DNA in some way, and then the DNA, in trying to fix itself, is going to fix itself incorrectly, and then that will cause the mutation. Here's a specific example of radiation, right? And so the radiation can either directly damage the DNA by breaking those double strands, right? So here's our double helix. It can be broken. The bonds holding it together can be broken, okay? And then the cell may die, okay? But it could also happen, right, that the DNA in either case is broken and then it is fixed by the cell. But like I said before, it might be fixed incorrectly, and then that mutation would then be preserved. But oftentimes the cell will know that it's been damaged, and then it will go through programmed cell death to protect your body from keeping that mutation. Here are some more examples. Here is albinism in a chimpanzee. So again, this could be possibly not too dangerous depending on the environment the chimpanzee is in. It looks like this chimpanzee has a friend to help it along. This is similar to the two-headedness, but more just two-facedness. Okay, so the second head has not developed fully separate from the original one. They're kind of partly fused together. And then here are some additional legs on a cow. So this would be a case in which there are certain genes that control, where your limbs are supposed to go, and then obviously how many of them are supposed to be there in the first place. And so if those genes get damaged or they get mutated, then this is what can happen: is you can have additional legs growing in the wrong place. And these genes are actually taken advantage of by scientists to do research. So they may purposefully try to grow things in a different place or express genes in places that are not normally expressed. All right, and so the question that I mentioned a little earlier, right? Are all mutations bad? Okay, and you might guess the answer is no. Okay, many mutations are in fact neutral. They don't really have any effect. And then some mutations you may consider to be good, right? Thinking back to evolution, right? Though we need mutations to create variation, and then that variation can increase fitness, for example. But let's take a look at why some mutations have no effect on the phenotype, right? Phenotype, if you remember, is the physical trait of an organism. And so here's an example. Here's our original DNA strand, okay? And this is where protein synthesis comes in. Okay, we've got TAC, CCG, okay, AGC, ACT, okay? And if we were to transcribe that into RNA, you can try to do that. Okay, it would be AUG, GGC, okay, UCG, UGA, okay? And then, so let's say we have a mutation of just one letter, okay, our G becomes a C, all right? And if we think about our amino acids, right? We're thinking about translation. We want to take our 3-letter codons and then translate them into amino acids. Okay, you can see if we change, well, we haven't changed this first codon, right? The TAC is still TAC, so that's not going to change. However, the CCG has now become a CCC. So if you take a look at a amino acid chart, you can see that these two codons, though they are different at that 3rd letter, actually do code for the same thing. Okay, so I'll let you take a look. If you can pull up a codon chart or wheel, you can try that for yourself. Okay, but ultimately what's going to happen is, let's say this codes a gene in this cute little puppy here. Okay, this mutation is not going to change the protein that this gene codes for because the amino acid is the same. And so, thank God, our cute little puppy, even though there may have been a mutation, still cute little puppy. Okay? And so this is why there are multiple codons that code for the same amino acid. It's a built-in kind of safety net for if mutations do happen, then there is a possibility that there will not be any kind of effect on the individual. All right, and so let's talk about types of mutations, right? There's different types of mutations depending on what's changed in the DNA. And so here is one point mutation, right? Point because a single base has changed. So one point has been changed. And this can either be a substitution, right? So our T becomes a C, it's been swapped. It can also be something's added or removed, okay? And so sometimes when the DNA is replicating or it's being repaired, an extra base can actually be accidentally added in, or it could be accidentally left out. And this is going to cause what we call a frame shift. And I'll explain that in one moment. But we can also think about mutations in terms of how they affect the amino acid, like I mentioned before, right? So this would be the original DNA codon, RNA codon, and then the amino acid. This is lysine. A silent mutation is the one that I was talking about earlier with the puppy. It may have changed, but it doesn't change the amino acid that it codes for. So the C may have become a T, all right, but it still codes for lysine. Okay, that's one possibility, and that would be kind of the ideal, right? If we have to have a mutation, we would hope that it doesn't have effect on the protein level. We can also have mutations that are we call nonsense, and so that is a mutation that causes the codon to become a stop codon, right? So remember there was those 3 special codons that code for the end of the protein. So if we have TTC and this first T becomes an A, then this is going to translate into a stop codon. And so if we were in the middle, this lysine was supposed to be in the middle of our protein, right? Think about proteins as being a chain of amino acids. Then having a stop where we were not supposed to stop is going to essentially cut that protein short, okay? And so, it was supposed to be 20 amino acids long, and now it's only 10, okay? So that's obviously going to affect its function. It's not the full protein. All right, and then finally we have missense, okay? So don't worry about conservative versus non-conservative, but missense means that the amino acid has changed, right? So originally it was lysine, but depending on which letter you've changed, it could then code for a whole different amino acid, right? So the protein hasn't been cut prematurely, but there is some change in what amino acid is there. All right, and here's just my explanation of those types of mutations written out, right? Silent mutation has no effect. The nonsense mutation changes the amino acid to a stop codon, which truncates the protein. Truncates means to cut short. And then missense is a change in the amino acid to a different amino acid. Okay, and so back to the frame shift, right? So I said earlier, you could either delete or you could insert a single base pair, and this is going to change the whole way that you would read the codons, right? Because the codons have to be read in 3s. So if you stick in an extra letter or you take one out, then you're going to have an issue, right? So let's say here we've got a set of codons and they spell out big old red dog, right? So I'm just going to use words to represent this. But if we get rid of this I, so this would be a deletion, well, we still have to read these letters in sets of 3, right? That's the rule. And now, I mean, I'll challenge you to try to say this out loud. We've, instead of big old red dog, we've got Bago, Ladur, Ed, Og. Okay, so it doesn't mean the same thing anymore, and it certainly in this case doesn't mean anything. Okay, and so you can see here this, it would have a huge effect on the entire protein because everything shifted, so everything changes. And then we could also have the same effect if we insert an extra letter, right? So let's see, I just throw in a random U here. Well, now we still got to read them in 3s, and so now we've got Bui Gold Dre Dog. All right, so it's almost like I'm inventing a language, but the point being is that it's going to change what it codes for, right? It changes the meaning. All right, and then another type of mutation we have is when we have big chunks of the DNA that have been changed, right? So the ones that I mentioned earlier were focused on just one letter, but a chromosome is made up of millions of those letters, okay? And we could have changes at the chromosomal level, right? So large segments of DNA are changed, right? So we could have, you know, a whole segment is deleted, right? So this could be thousands of base pairs that get deleted. We could have duplication, right? So it could be a chunk of thousands gets doubled in, okay? We could have inversion. So this is trying to show you that the B and the E segments have swapped, right? So the entire sequence has become changed in those areas. And we could also have something interesting called translocation, which is actually when 2 different chromosomes swap entire chunks, okay, that they're not supposed to be swapping, right? And then so you have genes that were supposed to be on this first chromosome have now jumped over to this other second chromosome, all right, and vice versa. Now that's definitely going to mess things up as well. So some examples of genetic disorders, I've mentioned some of these in my supplemental lessons already. So like cystic fibrosis, for example, color blindness. This is Huntington's disease, which affects the brain. These are all genetic. So we know what changed in the DNA to cause certain individuals to be born with these disorders. Cancer is also a big field related to genetic mutation because cancer is caused by certain mutations in your genes that control cell growth. Okay, and so I'll just quickly talk about it. But normal cells, if something happens to their DNA, like I mentioned before, they'll usually go through what's called apoptosis, which is the programmed cell death, because they want to protect your body from maintaining these mutations, right? They don't want your cells to still have this, so that cell will basically kill itself off, okay? It sacrifices itself. However, cancer cells, something's gone wrong with those genes that control this process, and so it's going to have this DNA damage, but it's going to keep dividing. It's going to keep growing, which is not good, right? And so cancer is one major example of genetic diseases. And so it could get to the point, right, of creating a tumor. That tumor can enter the bloodstream and go elsewhere in the body. That's called metastasis, okay? And then here is just another image of how that might happen, right? It could be radiation, it could be nutrient deficiency. So if you're not getting the correct vitamins and minerals, that can all cause something to go wrong with the DNA, and that can eventually cause some kind of cancerous growth. Okay, so that is the end of my mutations lecture. I hope it was interesting. And so the big takeaway for this one is just that things can change in their DNA, and because DNA codes for your traits by using proteins to express those traits, then mutations can therefore change those proteins, change those traits. Okay, so make sure that you understand protein synthesis, and then that will help you to get the most out of this particular topic. All right, so I will see you next time. I hope you're all doing well, and I hope you have a wonderful day.